Welcome everybody to a Striatech hosted journal club. My name is Thomas Münch and I am one of the founders of Striatech. In our journal club series, different scientists present their projects and data highlighting the use and the application of our products. Striatech is a dynamic biotech company from Tübingen in Germany. We specialize in neuroscience testing tools, in particular for vision research. The Striatech founders are all experienced neuroscientists in the field of vision research. Today, we welcome Jana Remlinger as our speaker. Her talk is entitled Anti-FcRn Treatment in Antibody-Associated Experimental Autoimmune Encephalomyelitis. Jana has studied biomedical sciences in Fribourg and in Bern, and she holds bachelor and master's degrees in the field of neurology. She has worked on motor function in the context of spinal cord injury and on neuroinflammation in the context of stroke and neuroimmunological diseases. Currently, she is pursuing her doctoral degree in Bern in the lab of Andrew Jan under the supervision of Anke Salmen and Volker Enzmann. Her PhD work is about antibody-mediated demyelinating disorders of the CNS. What she will present today is part of that project. There is a whole family of diseases that manifest in the demyelination of neurons. This has detrimental and sometimes horrific consequences for the affected individual. The purpose of any treatment is to stop this decline of health or even to reverse it. In basic research, in particular in animal research, a constant challenge is the tracking of disease progression. In many such diseases, also in today's example that Jana will talk about, the neurons in the optic nerve are affected by demyelination. As a consequence, the visual abilities of the animal are affected as well. This is why Striatech's optodrum can be used to monitor and quantify disease progression and the effects of treatment. The optodrum takes advantage of the optomotor reflex. The optomotor reflex is a visual reflex that makes the animal follow global motion in the environment. Experimentally, you can evoke the optomotor reflex with a rotating stripe pattern surrounding the animal, as seen here. Jana will show you today that her treatment was working. She was working with mice with a demyelinating disease MOGAD, and with measurements of the optomotor reflex, she could show that treatment with certain specific antibodies preserve visual function in her animals. Before you start, Jana, let me remind the audience that you will be able to ask questions throughout the event, and also to give a thumbs up to other questions. A quick word about asking questions. The interface can be a bit confusing. When you want to ask a question, the cursor by default is in the name field. Giving your name is optional, but you have to click into the question field before you start typing your question. If you do not click into the question field first, you will accidentally type your question in the name field. The Q&A panel on the right will stay available for a couple of days after the live event. So if you're currently watching a recording of the event and Q&A is still available, please make sure that you enter your question and your name so that we can get back to you with the answer. We are very much looking forward to your questions. So Jana, please go ahead with your presentation. Don't forget to unmute your microphone, and I'm very much looking forward to what you have to say.
Yes, thank you very much, Thomas. I'm just trying to share my presentation. One moment. So, is it okay?
Yeah, it's working.
Okay, great. So thank you very much for your kind introduction. And yeah, as you announced, I will present recently published work on anti-neonatal Fc receptor antibody treatment in an experimental animal model of MOG-IgG associated autoimmunity. So let's first start with some background on demyelinating diseases of the central nervous system in general. Demyelination is a common hallmark of CNS inflammation, and during this process, the myelin sheets that are insulating the nerve fibers are damaged. This leads then to nerve conduction problems. Probably the best known demyelinating disease is multiple sclerosis. But also other diseases have been described, and antibody detection in some patients has even allowed the identification and distinction of different CNS demyelinating diseases. From these antibody-mediated demyelinating disorders, we know that they cause severe neurological impairment even with a single relapse and one single inflammatory attack. As demyelination indicates the destruction of myelin, it seems plausible that myelin proteins have long been discussed as targets during the disease process. One such protein is the myelin oligodendrocyte glycoprotein, in short MOG. And this is found on the outermost layer of the myelin sheets, which you can see in this image. The demyelinating potential of MOG and MOG-associated immunity has been investigated and described in animal models. So here, MOG protein from different species has been used to immunize experimental animals subcutaneously, and they then showed extensive demyelination of the CNS. So thereby, the animal model for demyelinating CNS disorders, the experimental Autoimmune Encephalomyelitis, EAE, has been created. And later, Chris Linnington and colleagues have even described that this CNS demyelination could be aggravated after antibodies against MOG were administered to these immunized animals. This you can appreciate in these two images. So in the first one, there is the EAE model where you can see the inflammation of the myelin sheets, and more extensive demyelination can be found after administration of MOG-IgG in the EAE animals. In patients, the presence of MOG-IgG was also investigated, and it was associated with several diseases, like, for example, NMDA receptor encephalitis, adult MS, recurrent inflammatory optic neuritis, neuromyelitis optica spectrum disorders, especially in patients which were aquaporin 4 negative, and childhood multiple sclerosis, so cases of ADEM also showed MOG-IgG. This heterogeneous spectrum emerged possibly due to difficulties with antibody testing, because only since the cell-based antibody detection test against a full-length conformational human MOG is available, this disease entity, MOGAD, could be defined as an own disease entity. In patients diagnosed with MOGAD, the clinical presentation involves the optic nerve and the spinal cord, and such lesions then mainly lead to vision loss and paralysis in these patients. Here in these MR images, we can see that the spinal lesions often present as longitudinal extensive transverse myelitis. And here in the optic nerve, we see that there is inflammation, and also cerebral manifestations of the disease have been described. However, for this newly distinguished disease, MOGAD, the modifying treatments are lacking so far. The underlying pathomechanisms that have been described so far involving MOG-IgG are depicted here in this illustration. So the antibody enters the CNS parenchyma, where it then binds to MOG on the surface of the oligodendrocytes, and as I mentioned, especially on the outer lamella of the myelin sheets. This then leads to demyelination, and these histopathological lesions that result involve mainly T cells and B cells. Also macrophages and Ig deposition are described, and there is possibly complement deposition. So now that we know about the disease MOGAD and how antibodies against MOG are involved in the disease process, how can we tackle these potentially pathogenic antibodies? So when we think about IgG homeostasis in the body, an important player is the neonatal Fc receptor for IgG, the FcRn. As the name indicates, it has been first characterized in the transfer of passive humoral immunity from a mother to her fetus across the placenta, but it has become clear that FcRn is also present in other cells, and that by this, throughout life, it protects IgG from degradation and thereby prolongs the half-life of IgG in the serum. So here you can see that after endocytic uptake of the IgG from the circulation by, for example, endothelial cells, but also monocytes, the FcRn binds the IgG in the acidified endosome. This then leads to the recycling of this complex back into the circulation, and thereby the half-life of IgG is prolonged. However, in case of pathogenic IgG, this mechanism is quite unwanted, and we would rather like them to go into the lysosomal pathway and into degradation. So how can we interfere with this physiological function of FcRn? There are several ways described. So one possibility is the administration of high-dose intravenous immunoglobulins. These then saturate the FcRn and therefore lead to an increased IgG turnover. Another mechanism are recombinant antibodies with increased binding affinity for FcRn via their Fc region, the so-called antibodies that enhance IgG degradation, APDEX. And such antibodies have also been described that are additionally selective for a specific antigen. They're called CELDEX. But a more direct and selective approach to reduce serum IgG concentration is to specifically block the FcRn IgG interaction with monoclonal antibodies against FcRn. Therefore, this seems to be a promising treatment target in autoantibody-mediated disorders. So to this end, we have investigated such a monoclonal anti-FcRn antibody in a murine model of antibody-mediated CNS autoimmunity. So more specifically, we looked at the effects of this treatment on the visual system and the overall disease course in this MOG-IgG augmented EAE that I mentioned before and will describe now in a bit more detail. So these are our main methods. On the one hand, you can see the augmented EAE. For the EAE, we use an active immunization with a MOG-35 to 55 peptide. For this, the peptide is emulsified with complete spheroids adjuvants, and additionally, pertussis toxin is given to the animals. Then, shortly before onset of the disease symptoms, MOG-IgG was applied to drive the pathology towards the autoimmunity that is associated with the antibody. In general, EAE has mainly spinal symptoms, which are scored here on a 10-point scale, where 0 corresponds to no clinical symptoms, 2 to a complete tail paralysis, 4 to gait ataxia, 6 to a moderate hind limb paraparesis, 8 to tetraparesis, and 10 to death. The disease course usually consists then of one peak, followed by a slight remission of the symptoms. And as this score that mainly reflects motor symptoms without any visual component, we also assess the visual disease component by another mean. That is the optomotor reflex, which has been introduced already by Thomas. And so I will just quickly go over what we have measured here. So as presented, the animals are placed here on this platform that is surrounded by 4 screens on which this black and white stripe pattern rotates. And then the optomotor reflex is then assessed via this camera and via software which is attached to this camera. So when the threshold of the spatial visual acuity of this animal is reached, they do not recognize this pattern anymore, and thereby we can measure differences between the animals before and after treatment, and also between treatment groups. So on a timeline, our approach looks like this. We start with this functional visual measurement, OMR, at baseline, and then we repeat the measurement at the end of the experiment, so around day 20 after immunization. So day 0 is the day of immunization with the MOG peptide. As mentioned, day 10 is augmentation with MOG-IgG, and our treatment regimen is on days 7, 10, and 13, where we administer the antibody against FcRn or an isotype-matched control IgG antibody. But before we come to the treatment, we first did a re-evaluation of the MOG-IgG augmented EAE by testing this model against different control conditions. And here we could reproduce the findings shown earlier by Chris Linnington. So we can see that here is the clinical score, which I've explained before, and the days post-immunization. And we show that the MOG-IgG administration exacerbated the disease course compared to when animals that were immunized just got IgG as a control. Then both groups that received only sham immunization, so the complete spheroids adjuvant without a peptide, and did not show any disease symptoms, whether we gave them MOG-IgG or an isotype control. So then we could now use this model to study the effects of the anti-FcRn treatment. Sorry, I think my screen is stuck.
Do you want to try to unshare your screen and then share again? I mean, repair your PowerPoint and then share again? Oh, it's working again.
Good. So we have found that the anti-FcRn treatment ameliorates the clinical disease course of the MOG-IgG augmented EAE. So here again, we see that the clinical score, and we followed up the animals for 21 days from immunization. And the treatment group, here shown in green with the anti-FcRn, had a milder disease course compared to the control IgG group. And here I show you that we could also find that these clinical findings were reflected in our histological assessment of the spinal cord. So first, the percentage of demyelination of the spinal cord white matter was strongly reduced in animals that were treated with the anti-FcRn antibody, always here in black, compared to an IgG control. This was evaluated with an LFB staining, Luxolfas blue, which I show here below as exemplary pictures. So blue, the LFB is staining the myelin, and when we see here below that there's only the counter stain, which is PAS, is left, we see that there's the demyelination. Then we also investigated immune cell infiltration with immunohistochemistry stainings, and we could see that especially macrophage infiltration was strongly reduced in anti-FcRn treated animals compared to isotype control. The other histological outcomes, the T cell and B cell infiltration and complement deposition, did not differ between the groups. Our complement deposition was investigated with a immune fluorescent staining against the C5b29 complex. So, as mentioned, the visual system is commonly involved in MOG-IgG associated autoimmunity, and in our approach we also saw that animals had a decline in visual function over the course of disease when they received only control IgG, but the visual function was preserved in the animals that were treated with the anti-FcRn. As a histological correlate here, we again evaluated demyelination, immune cell infiltration, and complement deposition. And even though we did not see significant differences between the treatment groups, we found that there was pathology present in the optic nerve. And to follow up on this finding, we also evaluated the cell bodies of the neurons from the optic nerve, the retinal ganglion cells, as they could be a potential marker for the degeneration of the neurons. Here, however, we did not see any significant differences between the treatment groups. As an exploratory finding, we also performed a correlation analysis of visual acuity with demyelination of the optic nerve and also the number of retinal ganglion cells. Due to the low animal numbers in this part of the analysis, we pooled both treatment groups as they still have the same underlying pathomechanisms. And we could see that there was a correlation of visual acuity with optic nerve demyelination. However, it did not correlate with retinal ganglion cell numbers. So with this, I would like now to go over to the discussion of what I've presented today. So we have worked with a highly established experimental model of MOG-IgG augmented EAE that we could successfully reproduce. The treatment approach that we employed in this animal model has also been investigated in other autoimmune inflammatory diseases, for example, the experimental epidermolysis bullosa in mice, which is a rare skin condition, and also a mouse model of immune thrombocytopenia. And in both these experimental models, they could show that the anti-FcRn treatment lowered the circulating IgG. With these promising results, then it seemed plausible to apply this therapeutic approach to other antibody-mediated autoimmune conditions, as we did in our study, where we could then show the positive treatment effects on the disease severity and visual outcome of the animals. And we saw that the spinal cord pathology reflected the disease severity. However, for the analyzed optic nerve pathology, we only had a few animals investigated, and we therefore suggest further confirmation for this part. Surprisingly, a decline of the visual acuity of the animals was not reflected in the number of retinal ganglion cells. This might also be due to a short observation period of 21 days, because other studies have reported a loss of retinal ganglion cells in EAE animals more at later stages. Our study also leaves us with some open questions. So, as I mentioned, further confirmation of optic nerve and retina histopathology would be highly appreciated. And another open point is the exact mechanism of action of the anti-FcRn treatment. So the most straightforward explanation is the direct removal of MOG-IgG from the circulation. However, other mechanisms may also play a role. For example, FcRn is known to be involved in the modulation of immune complex presentation in antigen-presenting cells, as you can see in this image, for example, with this dendritic cell. In green, we see the FcRn antibody and how it is here involved in the presentation of immune complexes with IgG to, for example, T cells. And therefore, one could hypothesize that the modulation of this MOG-IgG response that is primarily T cell driven could also be involved. So taken together, I will shortly conclude what I've shown. We see that the anti-FcRn treatment preserves visual function and ameliorates disease course in MOG-IgG augmented EAE. The animals treated with anti-FcRn showed reduced macrophage infiltration and demyelination in the spinal cord, and no differences were found in the optic nerve regarding the treatments. Here, however, we have the quite low animal numbers. Thus, selectively targeting FcRn might still represent a promising potential therapeutic approach in MOG-IgG and other antibody-mediated diseases. However, the impact of MOG-IgG augmentation and FcRn treatment on the visual system needs a bit further investigation. And one last note on the patient situation. So currently, the application of a human FcRn antibody in a phase 3 study in MOGAD patients has been initiated. So at this point, I would like to thank everyone who was involved in the project, and especially Adrian Madaras, with whom I share first authorship, and my PhD supervisors, Anka Salman and Volker Ensmann. And thank you all very much for your attention, and I'm now looking forward to an interesting discussion.
Well, thank you very much, Jana. That was quite nice. Thanks a lot. We already have a couple of questions, but I invite our listeners and audience to ask more. All right, let's see. The first question, Jana, was about the treatment time point in which you inject the anti-FcRn antibody. You do that before, or you start injecting that before symptom onset? Is that something that's realistic? I mean, you would not know a patient needs that before you see any symptoms, right? Was this more of prevention, or is it an actual treatment that you're doing there?
Yes, thank you very much. It's a really relevant question. So the timing of the anti-FcRn application, so before the administration of the MOG-IgG augmentation, might really be a limitation of our study. However, the treatment was still given after the immunization phase. So the immunization provides this inflammatory background of CNS demyelination. Therefore, strictly speaking, it's not a prophylactic treatment. It is still after the disease process has been initiated. And of course, in patients, we can give them after.
Yeah. So this FcRn antibody, I mean, it's there for a reason. It serves its purpose in a normal person or a normal animal, and now you're interfering with that. Are there any adverse side effects that you would expect, either in your mice that you might have observed, or in the patients that you might already know about from the previous phase 1 and phase 2 trials, if they're already in phase 3?
Yes, so in the mice, we did not see any adverse events that we could observe or that were within our scoring of the animals. What we can expect is that because the whole IgG of the circulation will be lowered, so we can expect a hypogammaglobulinemia, and therefore there might be also a reduction of humoral immune response, and could possibly lead to more infections. However, in the animals, we really could not see any like obvious effects. And for the injection, so in patients, it has been reported in the trials that have been ongoing so far. So also in phase 2 trials with patients with immune thrombocytopenia and myasthenia gravis that are published so far, that the most common adverse events was a headache, and the most treatment related adverse events were mild to moderate. So there we do not expect so far that there were severe adverse events. Otherwise, would not go further to more trials and phase 3 trials, I think.
Yeah, this makes sense. You showed in the spinal cord that you don't see an effect on T and B cell numbers and infiltration. Do you have an explanation for that? Why there is no effect there?
Yes, that's true. So the main effects are really in demyelination and macrophage infiltration. And even though the EAE is mostly a T cell driven model, the overall number of the infiltrating T cells is relatively low compared to macrophages, which represent at this stage of the disease where we investigate the animals, so at the chronic phase, they are the main cell type that are in the lesions present. Therefore, the differences might be less striking in the number of T cells. And for the B cells, it's quite, they have been seen mainly meningeal, so it's quite difficult to really evaluate how the infiltration of the B cells is going on. You have to be really careful when you're preparing your samples, because you do not want to lose the meninges. So there are some methodological problems, let's say. However, in our analysis, we really did not see a difference, but the EAE model is also not really known to be a B cell model, so we did not expect a huge impact of the treatment on B cells there.
Okay, there's one question about the difference that you see in the effect of treatment on the visual function on one hand, which was preserved, and on the clinical symptoms, which were not completely ameliorated, right? So you had some clinical symptoms left. Why is vision helped so much better, do you think, than the rest of the clinical symptoms, the motor symptoms?
That's a really good question. So I could imagine that the visual decline is less severe, let's say, than the clinical symptoms. So when you treat the disease, we mainly treat the antibody component of the disease. So we still have the EAE model that is ongoing, and we cannot really completely reverse the whole process of the EAE. So we still see that there is demyelination and macrophage infiltration, even if it is ameliorated. And also within the normal EAE model, we often see that these animals cannot recover fully. So I would say the difference could also be that the optic nerve, in which we also did not see that much of a pathology or differences in the pathology, is less severely affected. So that we still have a treatment effect on the visual outcome, but that this might be then more effective because it was not that severely affected in the beginning. I hope this makes sense.
Yeah, I think it does. So there's one question. The question says if you only treated animals that had EAE symptoms, but since you started treatment before the symptoms start, I think you don't really have control about that. But let me phrase this more general. When you do this model with the MOG injection and then the immunization afterwards, is it very consistent in eliciting symptoms? Or when you measured the visual effects of your FcRn treatment, might you have been lucky, so to say, that you happened to find animals where you were not consistent in having disease effects? Or is this really a very consistent model?
So the model is quite consistent. We always have some animals that do not develop disease symptoms, but we then usually exclude them from our analysis. So if they do not develop motor symptoms, we exclude them because the EAE model per se did not work. And so that's why in this study we did not have any healthy animals. So all of them had really symptoms, and there was no bias between the treatment groups.
Yeah, there's one question about the location of where the treatment actually hits, so the FcRn receptor. Is that at all expressed around the optic nerve, or is it really a systematic intervention that you're doing there, or is it or does your treatment maybe act locally in the region of the optic nerve? So where is the FcRn receptor? Where is it expressed in general?
So in general, the FcRn is expressed on endothelial cell, but also on many blood mononuclear cells, so also monocytes and antigen-presenting cells, like dendritic cells, for example. So it's like really all over, also in the optic nerve, I would then presume. And our treatment is systemic treatment. So we did not like really see whether the FcRn is more blocked at one or another, in one or another region of the body.
Do you know whether when you initiate the MOG treatment, no, not the treatment, but causing the disease, is that dependent on the dose of MOG that is being injected?
So yes, I think several research groups use a bit different. So I think we're talking about the MOG peptide. I'm not sure. So if it's the MOG peptide per se that is causing the EAE, the different research groups have a bit different concentrations that they use or amounts that they use to immunize the animals, but mostly then the effects are over the groups the same. So and with more MOG peptide, of course, I think the reaction would also be stronger, but still to see that the animals develop the disease and have a reasonable clinical score, that is also, I mean, if the dosage is too high, you would also have a lot of animals that would have a really high score or would even not survive the EAE, which would really not be anything we want in our studies. If it's more about the MOG-IgG concentration, I think the same holds true a bit. So we used a concentration that is published, that was also used before. So we really use this model that has been shown to work, and we, as I showed in our slides, that we have tested it before. So we really see that there's an aggravation of the EAE, but still the animal is not so strong that the animals have really severe symptoms or we have to stop the experiments. I'm sorry, I cannot hear, I think.
Sorry, I have to unmute myself. Is there a long-term effect on the FcRn expression itself? So when you treat with the anti-FcRn antibodies, they're down-regulation or maybe up-regulation, who knows, of the FcRn antibody or receptor rather?
Yeah, that's a really interesting question. I actually haven't seen anything about or read anything about this, but I could imagine that it has not really. I'm really not sure about this, because I'm in the homeostatic situation. The IgG binds all the time to the FcRn, so it is always blocked and released and blocked and released again. So this would be like the physiological way, but yeah, I'm really not sure about it, but it's an interesting question. We have to look into that, I think. We did not investigate our model in detail about this FcRn itself.
Yeah, maybe there's a whole new project for you or for somebody else. I don't think there are any more questions coming in, so that was basically it. I want to thank you again, Jana, for the wonderful talk and also for your time that you took to answer the questions. I want to thank the audience for those wonderful questions. And I wish, there's one more new question coming in. Hold on, we still have some time. No, somebody was thanking you for that. More questions are coming, Jana. You cannot go yet. Have you done any comparison of the pathologies, phenotypes you've investigated in animals without EAE or with less traumatic clinical symptoms of EAE?
So I've not really compared different stages of EAE. So we have mainly these two treatment groups where we like pooled all the animals together, and sometimes they have a bit more severe and sometimes a bit less severe symptoms, which is quite common in EAE. So you're not always having like all the animals that have the same score on the same days. So we did not really stratify the animals by their clinical score. We have done one correlation analysis once for one experiment where we checked the clinical score independent of the treatment or the model, so with or without the MOG-IgG augmentation against the visual acuity, and there we saw that there was a correlation between also inverse correlation between the clinical score and the visual acuity. So the more severely diseased animals were, the less the visual acuity was. But other than that, we did not do any stratification.
What type of mouse strain do you use for the general approach?
We use 6 mice.
Like 6. Do you know about any differences in like how the mice react to that initiation of the disease in different mouse strains, or do you know if it has been used in rats, at that model, if it has been used in rats?
Yes, so the EAE model is quite well known and quite broadly known in different in different animals, for example, also rats, and also in different strains. And you usually get a different kind of EAE. So here we have like this one relapse with the slight remission of the symptoms. And if you use other strains, and you can also use other CNS peptides, for example, other myelin proteins like myelin basic protein, there you would have also different kinds of disease clinical scores. So you could also have a more chronic disease where you do not see any remission, or a progressive disease course. So they're really I think, more than 10 different EAE models out there with different strains and different animals.
Okay, I think this was really it now.
Okay.
Thank you very much, Jana, again. And again, thanks a lot to the audience. And I hope you'll come back to our journal club next time when we have a journal club later this year.
Yes, thank you very much for having me. It was really great opportunity to present my work here.
Yes, you're very welcome. Goodbye, everybody.